Welcome back to the Deep Dive. Today we're attempting to map the most intricate, complex structure we know, the human body.
It's an almost impossible task when you stop and think about it.
Right. I mean, just think about this massive number for a moment.
Mm.
You have an estimated 37.2 trillion cells inside you. Trillions.
And they span this unbelievable diversity of types and states, and every single one is constantly shifting based on, your ancestry, your age, your environment, everything.
It's just an overwhelming level of biological detail.
It is, and that very complexity has created a huge bottleneck in medical science. I mean, we've successfully cataloged over 100,000 disease-associated genetic variants thanks to global genetic studies.
Which is incredible progress.
It is, but here is where we get stuck. We often don't know which specific cells these problematic variants are actually active in, or what biological function they are disrupting.
So it's like we found a bug in the software code, but we don't have the operating manual to find which circuit board it's on.
That's a perfect analogy.
Without that cellular context, that precise location and function developing truly targeted diagnostics and therapies becomes, almost impossible. We're hitting a wall.
A wall of biological context. And that gap is exactly why we're talking about the Human Cell Atlas today, or the HCA.
Launched back in 2016.
Yep. It's this massive global scientific consortium that set an incredibly complex goal to create comprehensive reference quality maps of all human cells.
Yeah.
The whole idea is to fundamentally transform how we see health and disease.
Okay, so let's establish what this atlas actually is, because the concept borrows from two fields that seem pretty far apart: traditional map making and advanced geometry.
Right.
Let's start with the geographical analogy first. That seems a bit easier to grasp.
Think of it like a global map set. The HCA covers different organs: your lung, your kidney, your brain, which are like different geographies on a map. And for each organ, we need multiple views. We need to know which cell types are there, what states they can be in, like active versus resting, and crucially, where they are positioned relative to each other. It's a multi-layered map.
Got it. And then there's the mathematical analogy, the manifold concept. This is the one that might lose people. We need to simplify this.
Absolutely. Think less about, dense calculus and more about seamless integration. Imagine a curved surface like the Earth.
Right.
You can't map that perfectly with one single flat chart. You need dozens of local charts, the atlas, that each describe a small piece of that curve perfectly.
Okay, I think I'm following.
So in the HCA, each cell is a point in this high-dimensional space. The known cell types, they're the local charts that describe the diversity in one small area, and the algorithms let us move smoothly between these charts, describing how a cell differentiates without any, artificial breaks.
So it's about making sure all the little local maps fit together into one continuous, unified picture of the whole planet, so to speak. So our mission for this deep dive is to see how the HCA is moving past just collecting data and into 5 really powerful future applications that are set to be the engine of future medicine.
It all starts with the most foundational step, the cell census.
Taking the population count before we map the roads.
That's it. Before we do anything else, we have to systematically discover, catalog, and describe every single distinct cell type and state across every tissue in the human body.
And what kind of data is being used for this massive cataloging effort?
It's primarily data from single-cell and single-nucleus molecular profiling, mostly RNA sequencing so far. This is all being integrated into the first big reference map, HCA version 1.
So this is like the golden path assembly they did for the original Human Genome Project.
That's a great comparison. It's that same iterative process of just establishing a reliable, standardized baseline that everyone can build on.
And these early drafts, they're already giving researchers practical results, right? This isn't just a concept anymore.
Oh, they are, for sure. Take the Human Lung Cell Atlas version 1. By having this healthy baseline, researchers could suddenly identify rare cell types they'd missed before. They could also document the natural variation between healthy people, which is so key.
So you know what's normal variation versus what's disease.
Exactly. And then they could identify shared pathways across totally distinct diseases like COVID-19, lung cancer, and fibrosis. Now, a researcher can immediately compare their disease data to a robust, healthy reference. It just cuts down on so much work.
It must save years of effort. What about a case that isn't about a major disease?
The endometrial census gave us some remarkable insights into regeneration. The endometrium is the only adult tissue that can regenerate almost entirely without scarring, which it has to do every menstrual cycle.
Right.
The census helped pinpoint the specific cells responsible for that. And then, by comparing healthy and diseased tissue, it identified that two specific cell types are dysregulated. Their function is off in patients with endometriosis.
Which immediately tells you where to start looking for new treatments.
It points you right at the target.
Okay, this all sounds fantastic, but it's a global effort, hundreds of labs. I can just imagine one lab looking at a T cell and calling it one thing, and another lab calling it something slightly different.
You've hit on the essential challenge. It could be chaos, so we need rigorous, automated, and community-driven annotation.
How do you even begin to impose that kind of order?
Through four key mechanisms. The first is linguistic, the cell ontology. It's basically a shared hierarchical vocabulary, so everyone uses the same language to describe the same cell.
A shared dictionary. Okay, what's next?
The second and third are a community platform and computational learning. The HCA cell annotation platform stores the standard labels, but the really cool part is using transfer learning. Tools like that can take existing, trusted annotations and automatically apply them to new, unlabeled cells.
So it's like an AI auto-labeler.
It is, yeah. It standardizes cell identity really, really fast. And the fourth lever is just human intelligence, the biological networks. This is where domain experts come in and review the AI's work to make sure it all makes biological sense.
And all this standardization means the census can't just be a static database. It has to be functional, queryable.
Precisely. You need to be able to give it a new unknown cell and ask the atlas, what is this? Where does it fit? What are its active gene programs?
And that's where models like SEGPT come in.
Yes, foundation models like SEGPT and tools like Similarity are being built to manage these massive queries. The census tells us what the parts are, but it's only powerful if it's an actionable, searchable tool.
So we've got the census, the parts list, and the structure for standardizing it. But a cell isn't just its own genetics. It's also defined by its neighbors.
That's the pivot. A cell in isolation is not the whole story.
So we have to go from 2D to 3D.
Exactly. We need to know where cells are located and how they interact in a full 3D environment. This is why the HCA has to become a multimodal and multiscale 3D map, going from the tiniest intracellular parts all the way up to gross anatomy.
Let's talk about that word, multimodal, looking at more than just RNA. We're talking protein, chromatin structure.
All of it.
And what's fascinating is that these different layers operate on different timescales, right?
They absolutely do, and understanding that timing difference is it's crucial for figuring out what causes what in a disease.
How so?
I like to use this sports analogy. Imagine a football game. The chromatin, the DNA's organization, that's the head coach, it sets the long-term strategy for which plays are even possible. It's pretty stable.
Okay, the coach. Then you have RNA and protein.
RNA is the quarterback making the immediate play call. It's the leading indicator. It shows you what the cell is doing right now and what it's about to do.
And the protein.
The protein is the scoreboard. It's the lagging indicator. It shows you the result of the play that just finished. To understand what's going wrong, you need to see the coach's plan, the quarterback's call, and the score all at once.
And in developing systems, I hear that timing can actually flip.
It does. In development, when a cell is deciding its future, the change in chromatin structure, the coach's playbook, often happens before the RNA changes. Chromatin becomes the leading indicator there.
That's a critical distinction. So how are we gathering all this 3D information?
Well, techniques for in situ spatial measurement are advancing incredibly fast. We have this great example from human limb development. Researchers combined single-cell data with spatial profiling.
And what did they find?
They identified two distinct waves of muscle development, and by doing that, they could anatomically separate the genes associated with different congenital limb disorders.
And we're also connecting this tiny molecular data to the big picture, like MRIs and CT scans.
Precisely. The goal is to create one single common coordinate framework for the whole body, linking everything together.
This feels like where the AI component really starts to shine.
Yeah.
Can the AI actually fill in the blanks for data we haven't even collected?
Yes, and this is where it gets really transformative for clinical work. These models can learn a shared latent space across different data types.
What's the wow example here?
The prime example is research where models were trained on standard tissue histology, that familiar purple and pink H&E stain every pathologist uses, and single-cell RNA sequencing data.
Okay.
Then the model could turn around and computationally generate the single-cell RNA-seq data just from the H&E image alone.
Wait, hold on. So you're saying a doctor might, in the future, take a simple tissue biopsy, look at the H&E stain, and an AI could predict the complete molecular profile of those cells without ever running the expensive sequencing.
That is the ultimate goal. It makes advanced molecular diagnostics cheaper, faster, and way more accessible.
That's incredible. Okay, so moving from 3D space to 4D time, specifically human development.
Right. Mapping development is vital for understanding disease and regenerative medicine, but it's incredibly hard to study in humans. The developmental cell atlas community has made some big strides, though.
Like the prenatal skin atlas.
Exactly. That revealed how immune and non-immune cells work together to regulate how skin forms. And we now have crucial reference maps for the placenta, which helps us understand a healthy pregnancy.
But the 4D challenge is about more than just a series of snapshots in time.
Right. It requires capturing 3 maps at once. The fate map, what a cell will become, the lineage map, its family tree, and the morphogenesis map, how it moves and changes shape. It's a huge challenge.
We have the census, the 3D map, the 4D map. But all of this mapping is, well, it's pretty useless unless it accounts for human diversity.
This is a critical point. It connects the science directly to public health equity. We cannot create a useful map using a narrow subset of humanity.
And historically, genomic data has been overwhelmingly from people of European ancestry.
Nearly 90%. So if the reference maps only reflect that one population, any treatments we develop from them will be less effective for everyone else.
So to get real statistical power, the HCA has to scale up massively.
We need at least a 10-fold increase in profiled cells and tissues, specifically through big collaborations across Asia, Africa, and Latin America. This isn't just about, being thorough. It's central to personalized medicine.
Are we already seeing examples where including this diversity is paying off?
Absolutely. The goal is to connect subtle genetic variations directly to cell type specific effects. We've already seen this help identify splicing regulation in autoimmune diseases.
And I read about one with breast tissue.
A really striking one. Studies on breast tissues from women of different ancestries revealed variations in gene expression that are directly linked to ancestry-dependent differences in breast cancer rates. It's proof that diversity is a direct path to understanding disease.
Okay, so we have all these elements. But the reality is we can't profile all 37.2 trillion cells in every person their whole life.
Yeah.
At some point, the computers have to take over.
Computational inference is the only way to make this practical. Machine learning has to be used not just to analyze the data we have, but to predict the likely range of cell states from an individual's unique genotype.
To fill in all the gaps.
All the gaps. The ultimate map would be able to predict your tissue's behavior based entirely on your personal genetic profile.
Which brings us to the final goal, the foundation models for biology. This is where it all comes together.
This is the HCA moving beyond being the Google Maps of the human body toward becoming the periodic table of our cells.
I like that. Not just a map, but a predictive theory.
Exactly. These foundation models are vast, pre-trained AI models that can learn the deep structure of the data and then generalize from it. Models like Genoformer and SEGPT are already showing huge potential.
What can they do right now?
They can seamlessly integrate new data, rapidly annotate novel cells, and most importantly, predict the outcomes of perturbations.
So you could ask it.
You could ask the model, if I administer this drug, what is the most likely healthy state this diseased cell will transition into? It shifts biology from observation to prediction.
What's the most ambitious promise here?
The ability to predict entirely new, unobserved cell types, or to generate predicted cell states based purely on a novel genetic profile. If these models succeed, it means they've learned the deep, universal language of biology.
And all this work is already having an impact on current biomedical research.
Oh, absolutely. The methods developed by the HCA community are integral to disease atlases and clinical studies right now.
There was a recent example with SARS-CoV-2, wasn't there?
A really insightful one.
Yeah.
By comparing the nasal cells of people who got infected versus those who were protected despite high exposure, the atlas revealed distinguishing features in the protective group. Specific immune cells, different barrier compositions.
Which directly helps us understand how the virus gets a foothold.
And it informs our understanding of innate immunity. That's the power of having a healthy reference map. This whole endeavor, the census, the 3D map, the 4D map, and the final foundation model, it serves as the gold standard baseline for everything that comes next.
It's the framework for future monitoring, diagnosis, and treatment.
The goal isn't just a detailed picture, it's a predictive engine.
So we'll leave you with this final thought. If these AI foundation models can truly learn that biological language, the fundamental rules that govern our 37.2 trillion cells, what is the first major disease mechanism you think we will finally be able to decode and universally predict using this complete cell atlas? We look forward to seeing what you uncover next.